The overall goal of the following experiment is to measure the conformational changes in membrane proteins in their physiological state. This is achieved by site-specific labeling of membrane proteins to measure their LRET lifetime and thus the distance between the two sites. As a second step, a ligand is added, inducing a conformational change that can be measured. Next, the appropriate protease is added to cleave one of the fluorophores and obtain the background signal. Ultimately, the change in the fluorescence lifetime from the resulting resonance energy transfer can be measured to measure the conformational changes that occur in the protein structure.
The main advantage of this technique over existing methods like X-ray crystallography is that with this method we can measure the dynamic changes of proteins in their physiological state.
Though this method can provide insights into ionotropic glutamate receptors or acid-sensing ion channels, it can also be applied to other types of proteins like nicotinic acetylcholine receptors.
We're going to show the technique of luminescence resonance energy transfer, particularly in the context of studying conformational changes of membrane proteins in mammalian cells. My students Dolino and Swarna Ramaswamy will be demonstrating the procedure.
Before starting the experiment, it is important to choose labeling sites that are able to reflect the possible conformational changes within the protein. If possible, use the crystal structure of the protein or of a homologous protein to help make this determination. Choose residues such that the side chains of the selected residues are surface exposed and accessible to the fluorophores so that the protein can be labeled.